Hello, and thank you for joining us today. Welcome to our webinar, Building a Biotech, hosted by Benchling. My name is Aaron Stager, and I'll be moderating today's program. We're very excited to introduce our new Benchling for Startups package, and to do that, I'm joined by three of our startups experts at Benchling, Sean Kamal, Tom McCrory, and Sean McCormick. All have worked with hundreds of biotech startups to help them scale. So what will you take away from today's program? After spending an hour with us, you'll understand how the new Benchling for Startups program can help growing biotechs to scale effectively. You'll get a live look at our platform configured with the representative startup use case. Lastly, you'll have the chance to pick the brains of our speakers about all things startups and data during the Q&A session. Now, before we get started, there are a couple housekeeping items I'd like to mention. First, please feel free to ask questions at any time in the Questions tab on the right panel of your display. We'll answer as many as we can during the Q&A session and follow up with you individually for any that we don't get to. Second, we'll be doing some audience polls throughout the presentation, so please participate. And lastly, we're recording this webinar, so if you miss anything or you're interested in forwarding it to a colleague after the event concludes, we'll be sending the recording out via email. First, I'd like to start with a poll to provide some context for our discussion today. Currently, how does your organization keep track of research data? I think we still have a couple more votes coming in. Awesome. Yeah, it looks like we've got a lot of paper notebook users, folks using Google Docs, ELN software, even some Benchling users, so very excited to see that. Just goes to show that everyone is using a variety of different solutions. All right, I'd like to turn over to Sean to introduce our new Benchling for Startups package. Sean, take it away.
Thanks, Aaron. I want to take a minute to set the stage before we jump into Benchling. Biotechnology is fundamentally rewriting the way we live, from human health and agriculture to materials and energy. As the pace of scientific advancement continues to accelerate, more multi-dimensional data sets are being generated, and the sheer amount of data being created within life sciences research is an order of magnitude larger than ever before. It's important to get data management right at the beginning. Centralizing and standardizing data is crucial to decision-making. This data will help biotechs raise additional investment, file patents, and establish credibility with large pharma partners. Scientists lack a unified platform for experiment tracking and data capture, making it difficult for them to get a global view of their progress. Aggregating information across several experiments is nearly impossible, and key decision-making comes at a cost. If a company is deciding which of their drug candidates to spend millions of dollars to put through clinical trials, it's crucial that their data is accessible and standardized in a single, secure location to inform those decisions. Biotechs spend a lot of time wrangling with these data problems, trying to consolidate across scattered sources, just as we saw from spreadsheets, emails, handwritten paper notes. Hours of productivity are lost, hunting down information instead of focusing on what really matters: science and innovation. We've spoken with over 500 companies, small, medium, and large, and all of them are spending the majority of their time on data management because they don't have the proper tools to manage their data. And that's why we built Benchling. We work with hundreds of early-stage startups, and I want to take a few minutes to share some of the key reasons why startups choose to partner with Benchling, because we have everything that a biotech company needs to get off the ground with room to grow. Benchling is a unified platform with everything in one place, so scientists don't need to jump between different softwares. Data has been centralized and standardized in one place, so scientists can easily find what they need. Benchling was built for collaboration, so sharing data and context between teammates is straightforward. And we work with customers across many scientific verticals, and Benchling can accommodate many different types of science. Benchling will help build a strong R&D foundation that will help startups innovate faster, make smarter decisions, and grow at the pace that they need. The benefits and impact on bringing on Benchling as a foundation will enable companies to get going quickly with our implementation specialists to help companies collaborate, record, measure, and analyze consistent data from an early stage. This strong foundation will continue to serve companies as they continue to grow. I'd like to take a brief pause here to ask another poll question. What is currently top of mind for you as your company grows? So let's take a look at the results. So collaboration and tracking samples seems to be the most important piece. I think this will be really interesting to see, as I'm now going to hand it over to Tom, who will walk us through the demonstration agenda today and how that maps to some of these Benchling capabilities.
Thanks, Sean. So, hey everyone. So for our product tour today, I'll be taking you through 3 different scenes. We're going to start with the notebook, where I'll cover the basics of its use in the context of designing a plasmid. Next, in preparation for an in vitro experiment, I'll perform a search for that plasmid that I'd like to use and review how I manage it both at the sequence level and at the registry level. And then finally, I'll utilize a new notebook entry created from a template to capture some experimental data from our newly created samples. All right, let's get started. So for our webinar, I've created this fake company called Innovate Bio. And as a scientist there, I'll be completing a plasma design notebook entry that's about ready to submit for review. So here, the notebook is an easy to use, flexible scientific word processor. So within it, for those that aren't familiar, I can insert things like Excel-like tables for experimental planning and design, well plates for mapping out experimental conditions, attachments, and then a number of different structured type tables to interact with other areas of the platform. And all of these tools are really built for scientists in mind. But really, more than just a word processor, I've got a number of other modern elements, like the ability to @mention my colleagues as you would in like a social media platform. So here I'll do that. So I'll just @mention an auditor and send a quick note. So now my colleague here has just received an email with my note and a link directly to this notebook entry, really allowing me to asynchronously collaborate with them all in one place and streamlining this kind of communication. Additional kinds of @mentioning are things like my backbone here for the plasmid, these gene inserts, and then the product or that plasmid that the new plasmid I've constructed here. So other kinds of collaboration I can do within Benchling. Say I'm working with an external collaborator like a CRO or another partner. I can generate a read-only link for them here. So if they don't have access into Benchling, I'm able to share what I've been working on or maybe the results of a particular experiment with them. All right, so next, as I scroll down here, you'll notice that I have a rendering of my plasmid that I've constructed. And from its inception, Benchling is designed to be sequence aware. So that means that my notebook entry here is intelligently linked to the sequence here of this plasmid that I use on a routine basis. So clicking on any of those links that I created above with that mentioning or into this map will bring me directly into the molecular biology tool. But to get a better understanding of the flexibility of the platform, I want to look a little bit more how I capture some information within this entry. So as I come down here, you'll see an example of one of those Excel-like tables, where when I click into it, you'll see I use this to calculate a Gibson assembly master mix with some mathematical functions as you might use in Excel. And use this again, like for experimental planning and design. As I continue down here, I've put in an attachment into my entry. So this is here is a PowerPoint, but it could be any kind of a file, including something like for other software like a Flojo or any kind of graphing files that you might use. And because this is readable, I've surfaced a image of that PowerPoint. And because this annotation is machine readable, I can actually search on it. So if I were to search here based on this ID, I'd be able to pull it up based on the annotation within the attachment. All right, so once I'm about ready to submit this for review here. So since I'm able to do this, it's as a main part of our 21 CFR Part 11 compliance. And to be able to do this, I'm going to come here to our information pane, where I can then send this out to review to an auditor. I'll have the ability to leave a comment and create a review. And because I'm using a single sign-on, I'll also have the ability to authenticate my user identification. So if I were to do that, my auditor now would receive an email alerting them to this entry ready for review, as well as a link to that that entry, where they would then come into the review pane and have the ability to review my entry based on some maybe predefined criteria. All right. So now that I've completed this plasmid construction and entry, I'm ready to move on to the next step of my process and purify an antibody. First, though, I'll need to perform a quick search to find that plasmid and identify the inventory. To do so, I'm going to come over to our registry here. If I take a more expanded view, I'll see that although on the surface, this may look like one of your Excel spreadsheets, the registry really allows me to perform some more advanced searches across a wide range of items that I work with. It also helps to maintain organization within the lab, but also create linkages. So between my plasmids, genes, parent plasmids, cell lines, and the like. So here I'm most interested in a plasmid. So I already have this down selected here based on plasmid. But since over time, I'm going to have hundreds, thousands, tens of thousands of plasmids, I'm going to need to be able to search and down select even further. So I'll do so using a simple filter. So here I'll add a filter for one of the fields on our plasmid, namely a gene of interest. And I'll select that gene of interest after I apply that. All right. Now from our view, we've gone from a much larger number to now just two different plasmids that contain that gene of interest. So here you'll see a wide range of tags associated with each one of these. So things like standard tags, like their ID, the name, inventory, which we'll get to in a minute, as well as things that are linked to here, like that gene that we use to surface, or a promoter, or that parent backbone. And those that have sequences here are actually the actual sequence that I can click into and be driven directly to that sequence if I wanted to inquire more about it. So as I had mentioned, I'm mainly interested in the inventory. We're going to look a little bit more at the inventory in itself in a minute, but I do see right off the bat that I do have a vial of this in inventory that I'd be able to draw from and not have to worry about having to create one on my own. So I had mentioned earlier that Benchling is sequence aware, and because of that, this isn't just a piece of record. It also contains that DNA sequence associated with it. And by clicking into that here, I'm now going to be navigated directly into the molecular biology tool, where I can see a wide range of different types of things that you likely are familiar with. So things like annotations, cut sites, primer design tools, creating alignments or in silico assembly tools, both individually like I used here, but also in a bulk combinatorial fashion. It is also possible. But being able to design plasmids and proteins is really just step one. What I really need to do here is to track a lineage and the experimental context of this plasmid that I've created. And I do this by assigning those tags, which you've been looking at some. So to view those tags associated with just this plasmid here, I can click into the metadata tab. And by clicking there, I now see those full set of tags that we have just been looking at. And if I come to the bottom here, I'm actually also able to view things like relevant items. So relevant items being the other objects that I work with in the lab, or even that experimental entry that we were just in, this plasmid design and construction notebook entry. So you can really see how there's complete lineage and tracking between your entry, but also those objects, this being a plasmid. So here we've created this plasmid for the registry, but now what I need for the next step of my experiment is a way to create new samples, put them in inventory, capture experimental results, all in an efficient way. So to perform all of those, I can do that directly from my notebook entry, eliminating any needs to kind of click between systems or tabs within my Google Chrome, as a result, eliminating any chances of errors. So now we'll jump to that notebook entry that's in progress that I've created from a template, where we'll purify that antibody and capture a basic experimental result against it. All right, so here's that entry here, this protein production and testing. All right, so now that we're here in our experimental entry, This again was created with a template. So in large part, this came in pre-populated. But through use of those insertion tools, I'm able to customize and tailor this if my workflow is slightly different or had a more novel experiment. These can be created quite rapidly and ad hoc if necessary. All right, so we'll scroll down a little bit into my experiment, and you see that I've utilized this first registration table here. This is one of those structured tables that I can interact with the registry directly from the notebook. Within this table, I've defined a couple attributes, namely the parent cell line, some HEC 293s, and a plasmid. So this is that same plasmid that we've kind of been looking at along the way. Once I completed that information, I submitted that to my registry. So that kind of brings us to our current state. Now we need to purify this protein. So to do so, I'll populate this table here through looking up our structure table that's just above. So we'll pull in that parent cell line, and I'll have 3 different samples, say 3 different preps of this protein. Can define some more attributes about that protein. I say it's an antibody, maybe it's an IgG, and these are all the same. So we'll populate the rest. And when we've completed those attributes, I'll submit that or commit it to our registry. So once created here, there's a unique identifier or a registry ID. And I've created a basic lineage here that you can see. So I've got the protein here, but that's linked to our cell line, which is then again linked to that plasmid of interest. So where we can track back all these different pieces. All right. So we now want to say store a couple of these aliquots in inventory. So to do that, similarly to above, I'll add those from a structured table. So we'll kind of work these in tandem. I'll pull in my protein and just store, say, 1 vial of each. I'll select a box for my inventory hierarchy. So you see the representation here. We'll put them all in that same box and then ask Benchling to look into that box and assign a position based on a next available. All right, so you see all of these aren't necessarily sequential. So if I was to maybe do this on my own or kind of wanted to look in there, I can then click into that box to get a better view of what's in there. All right, so in clicking into that box, now I have a full representation, a physical representation of what's in that box. And I can do a whole wide range of inventory management tasks from here. I can see what each one of these containers contains, as well as some that are yellow here, which indicate that they're either reserved or checked out by another individual. And kind of scrolling through them, I've got a little comment on who had used them. In this case, they're myself, but you get the idea. So once defining this box, I'm able to then, I'll come back to our notebook entry and we'll transfer those vials into our box. All right, now we've transferred them into our box. Here we've foregone in completing some of this information, but you could complete the volume and concentration for these samples, as you saw with some of those others within that box. So far, what I've done here is I've created a new cell line, purified that antibody, and transferred some samples into inventory. But now we're ready to capture some of that purification data within Benchling. So, and the reason we're doing all of these is to, in particular, the transfer into inventory, so we don't have to hunt and search for these items in the lab, especially those that are cell lines. Nobody likes diving into doers. So here we've got some HPLC report that I'm going to upload. We could add samples like we had kind of done with those previous ones, but here we're going to actually import it from a spreadsheet. This could be directly off an instrument or something you've already parsed. Okay, so now I'm able to drop this directly into my entry right here. one moment and I'll pull it in from my desktop. All right. So next here, Benchling will allow me to verify these columns, and we will import that data directly into Benchling. I also have the ability, not just raw files like this, I can also upload files associated with this data. And in this case, maybe it's a silver stain gel of my purified protein. All right, so all these data are now linked and aggregated directly to that antibody and corresponding cell line that I'm interested in. So if I ever have any question about what those data are, I can either click into that protein purification, where I can view the metadata that we captured, the containers that we have in inventory, and that result captured on that protein. We do have a few different proteins. So if we want to see even aggregated even more, everything created with the cell line, and clicking into that cell line, I can also view all of those results just captured again that were from that cell line here and have linkage back to that entry. So hopefully here you've been able to see how these data are bidirectionally linked and aggregated all in one place. But for the end user, me in this case, I have a streamlined approach for capturing all of these data in just one place. So that wraps up today's product tour. I want to do a quick check-in with the audience with a poll question before I break. So based on what you've seen today, would you be interested in learning a bit more? We'll give a couple seconds for folks to answer here. And as you answer, I'd just like to thank you for your interest here, and we'll be sure to follow up with any of you that answer yes here shortly. Okay, so now I'm going to pass it back over to Sean and provide some closing thoughts before our Q&A.
Thanks, Tom, for walking us through the platform. As you've just seen, Benchling's startup program provides everything a startup needs to be successful today, and as they scale up, Benchling will be able to grow with them. By partnering with Benchling early, you're laying the foundation for the future. Any startup can feel comfortable knowing that as their science evolves, Benchling has additional functionality that will evolve with them to manage workflows across large teams. We all know how important data is to biotech companies, and having a centralized and standardized data foundation will be instrumental to support future IND filings, fundraising activities, and large pharma partnership. Hundreds of biotech startups have partnered with us through their growth. Zymergen is a great example. They started with us just a few years ago as 3 scientists and built their entire organization on Benchling. Since then, they have grown to 250 scientists using Benchling and recently went public. At Benchling, we know what it takes to help companies grow from startup to scale up and beyond. And that's why 99% of companies that purchase Benchling stay and grow with us. We're proud to work with hundreds of early stage startups, many of which switched from paper notebooks, email, and spreadsheets. Others chose Benchling because their scientists were so frustrated with other software that did not have the functionality to design, connect, and track their experimental data to meet their needs. They chose Benchling because they knew that their previous systems couldn't support their company's growth the way Benchling can. And we are excited to continue supporting the biotech industry. If you're interested in learning more about the startup program or have questions about pricing, feel free to contact me directly at sean@benchling.com. Now I'll pass it over to Aaron to start the Q&A session.
Thanks, Sean and Tom, for the great presentations. I'd like to open things up for the Q&A session. It does look like we have at least one question in the chat and a couple of kind of frequently asked questions that we'll run over as well. So yeah, let's get started. I'd like to bring Sean, Tom, and Sean McCormick on stage. So first question here from Risa about Excel. Oh, can you drag and drop from Excel? I think this is a good one for you, Tom.
Yeah, absolutely. Yeah. So that's exactly what I did. You couldn't see the file. It was an Excel file, either a .csv or your rich standard Excel files. So you can drag and drop or simply copy and paste into those tables. They function very much as you might expect.
Awesome. Thank you. And then from Kennelly, what is the average size of the startup companies that we work with? I think Sean Kamal, you could take that.
Yeah, it's a good question. So Benchling works with companies of all sizes. We have scientists, as we fondly refer to as 2 scientists in a molecule, all the way up to some of the largest companies in the world that have thousands of scientists. The average for startups is somewhere between I'd say 20 and 30 scientists. But this program is geared toward small companies that are either venture backed or just spinning out of academia, and they're just running their first experiments. So we have offerings and options available for everyone.
Perfect. And as folks might be kind of brainstorming, okay, here are a couple more coming in from Alexandros. How would you use notes versus protocols? Why do protocols have limited options compared to notes? Tom, I think this would be a good one for you.
Yeah, absolutely. So, we can attach a number of things. So within Benchling, it sounds like you may be familiar with the notebook itself, you can utilize protocols generated within Benchling that have a more structured format to them, materials, methods, kind of a structure. Conversely, utilizing experimental, entries a little bit more flexibly as protocols and linking them at the top of your entries. It really depends on the kind of flexibility you need within a protocol or a method, what approach you might be wanting to take.
Great, thanks, Tom. We have a question coming in about AWS and how does Benchling integrate with AWS? Tom, would this be a good one for you?
Sure. Yeah. So we're hosted in AWS, so it's a cloud-based platform, as you saw there, and we utilize a standard architecture there, things like S3 bucket for attachments, and as well as as part of our developer platform, things like the event bridge and things of that nature, depending on what kinds of needs you have.
Great. Coming in from Kennelly, do you provide ongoing customer support? And if so, which kind? I think this would be great. Sean McCormack, team lead on our professional services, to answer.
Yeah, happy to take this question. And hello, everyone. Sean McCormack, team lead on professional services, Aaron said. We do provide ongoing customer support. So I think the thing to note is that once you purchase Benchling and you sort of begin your journey with us, we'll start by getting you spun up. You'll have an implementation lead who's going to spend the next 2 to 6 weeks with the startup program, getting you live on Benchling, getting the platform configured, and making sure that your end users and your team are really trained up and know how to use the platform, and it's designed in a way that's compatible with your scientific workflows. Following that, you have 2 types of ongoing support. We always have a support team that's available either by emailing support@benchling.com, or in the Benchling app, there's actually a chat window, and you can just have an instantaneous chat with our team. They'll respond within an hour or so during business hours, and just kind of ask your questions there about the things that you need. And then the other option that you'll have, and this comes with every one that purchases the platform, is customer success. And so customer success is really geared towards your long-term success. You'll have a customer success lead or a group that's looking after your account, making sure that you're notified about the newest additions to the Benchling platform, if we release new features or tooling, and just over the life cycle of your time with Benchling, making sure that you're getting the best and the most out of the product.
Awesome. Thanks, Sean. Another question coming in from Risa. Is there a task manager type of function, Tom?
Yeah, absolutely. So, what I showed you today is kind of the core product that we offer to startups. We have some other functionality within the product to manage things like requests, and within those, there are tasks embedded, as well as a really wide range of upcoming features that can start to address those things. But that's definitely generally outside the scope of what we would encourage for a startup at this.
Awesome. And from Chris is asking, is there a dark mode option, Tom?
Yeah, so I would take it in terms of like flipping it from a white screen to a black background. Currently, there's only this one way to view Benchling, just in this view.
Great. And another question from Risa. Does your molecular biology function seamlessly integrate Genius files and export in a format that is compatible with Genius or SnapGene? And I believe we had another question in the chat about how Benchling integrates with Genius or SnapGene. So if you could shed some light on that, Tom, that would be great.
Yeah, absolutely. So any of that, like that plasmid construct from Benchling, you can either ingest Genius files, SnapGene files, or and conversely export things in several formats: GenBank, FASTA, RDF. There's also vector plasmid map. So there's kind of easy to kind of move between those two without issue.
Awesome. Thanks. It looks like we have the last unanswered question here. How do we find out what integration support you have? And thank you for letting us know there's some questions in the chat. I've seen those. Yeah, Sean McCormick, would this be a good one? Or
I think it's appropriate for either myself or Tom. So in terms of integration support, I think it depends on what type of integration you'd like to set up. We do have a developer platform and public APIs, which you're able to access and write custom scripting to basically hit our API endpoints and either add data to Benchling or pull data from Benchling. If you want to maybe elaborate on the type of integration you're thinking of in the Q&A, we could go into that a little deeper. But broadly, we do have a developer platform, and you are able to write custom scripts to hit our APIs. And there are API endpoints for most of the things that you can do in the UI so that you can do them programmatically as well. And Tom, maybe you want to add anything there that I missed?
Yeah, no, exactly. I mean, I think we integrate with a wide range of instruments, other databases, other types of systems. So it really is based on your use case. Yeah.
great. And I think we have one question that was just generally in the chat. I don't think we can surface this one on the screen, but a question for you, Tom, about reformatting table column height and width effectively. Arn is having some difficulty making tables printable in the case that you'd want to print it out for any reason.
Gotcha. Yeah, I mean, you can use the sliders to drag and drop and reformat table column widths, and text will wrap within it. So that would be the main go-to in terms of reformatting those tables. Yeah, I'd be interested to know more, though. We could certainly assist you with that if it's an ongoing problem for you.
All right. I think we answered all of them. Of course, if any questions come up after the fact, you can always feel free to reach out to us, and we'd be happy to chat about them. Well, that concludes our session for the day. Thank you for spending the past looks about 30 to 45 minutes with us. I hope it provided an engaging overview of how our new Benchling for Startups package can help your company to scale effectively. If you're interested in learning more, don't hesitate to reach out to Sean at sean@benchling.com, or visit our website at www.benchling.com and request a demo at /request-demo. Until next time, thanks for joining.